In this chapter, we're going to discuss the major chemical processes that occur inside of microbes, and a lot of these will be the same chemical processes that occur in multicellular organisms. So kind of as always, that's sort of my background, so I'll sort of probably be using some examples from that and also tying it in because you guys all will have learned um, some of this stuff already. So metabolism. Metabolism comes from the Greek word change, metabol, change. And metabolism is all of the chemical reactions that occur inside of a cell. So a chemical reaction, as you remember, is creating or breaking a bond, and metabolism includes both of those. Now, those, what I just described there, breaking and making bonds, have special names. Anabolism, sometimes called biosynthesis or other things like that. And it's synthesis, putting things together, which means we're building molecules by putting atoms together or other molecules together to make bigger molecules. Um, but the point here is that we're making a bond. And if we're making a bond, it's anabolism. So you may have heard the term anabolic steroids. Those are steroids that cause anabolism. In other words, they make bonds, they build. Um, so for a bodybuilder, they take an anabolic steroid to help them build muscle. So to kind of tie that in a little bit with something you already know. Now, when we make a bond, as you remember from one of the previous chapters, when we make a bond, it requires energy usually. We'll just say it does. Catabolism then would be the opposite of anabolism, and this is where we're breaking bonds and creating smaller molecules or breaking molecules up into individual atoms. Now, when we break a bond, it's a decomposition reaction, and that releases energy. So why do we need to metabolize things? So we have to build molecules because we use them inside the cell. For example, if you need to build parts of a cell or build structural components such as collagen fibers or elastin fibers in a multicellular organism or inside of a microorganism, cytoskeletal proteins, um, actin is an example. And as we said, energy is used, and the form of energy that is used is ATP almost all of the time. Now, metabolism also degrades macromolecules, because remember this, that's catabolism, into smaller molecules, and we do that to get energy. This is the reason, and we'll discuss this in detail, this is the reason that we consume uh, glucose and other types of energy sources. It's not the only reason we consume food, because we also consume uh, for the nutrients, for the building blocks, but we need energy and building blocks. So when we break those bonds apart and decompose them, it releases energy, and then we can take that energy and do things such as create an ATP. So this figure in your book just kind of tries to illustrate some of the possibilities of why we metabolize. So on this left-hand side over here is catabolism, and it's breaking down a glucose, running it through these reactions here, which we'll discuss. And these don't all happen in the same organism, um, and we'll talk about that. Some of them do. Anyway, it yields energy. And what we end up with are smaller molecules. Now, this also may be confusing because it looks like we always take glucose to create precursor molecules and then we do use them to do this. That is not necessarily true. These precursor molecules can come from all over the place. So maybe sort of in your mind separate this side over here from this side over here. Although it could happen this way, it doesn't necessarily have to happen this way. But examples of precursor molecules: pyruvic acid, acetyl-CoA, glyceraldehyde 3P, and thousands and thousands and thousands of others. Okay. Over on this side then is all anabolism, everything that we see in blue. And the kind of things that we anabolize is we put these precursor molecules into things such as amino acids. And when I say we, I don't really mean we. I don't even know what I mean when I say we. Um, Organisms can do this. And they use these building blocks to, or these precursors to create things like amino acids or simple sugars, or nucleotides to be made into nucleic acids, or fatty acids to be made into fats, and so on. So we put them together and make what we call macromolecules. So amino acids make proteins, sugars make bigger sugars, or in this case, they put proteins together with sugars and make peptidoglycans. Um, Nucleotides make RNA and DNA. Fatty acids are put together to make fats. And then we can put these macromolecules together and actually create cell parts, such as organelles in the case of eukaryotes, um, cell membrane in the case of all organisms, and so on. So we're going to discuss enzymes for quite some time now. Enzymes are what we call catalysts. And you've probably heard that word. It's sort of a euphemistic word that we use in the English language. You may not really know what it means. Um, but a catalyst sometimes is used to because it triggers something, and that's sort of what it's being used for in this sense. But the definition is it increases the rate of a chemical reaction without A, becoming part of the products, or B, being consumed in the reaction, and C, the need for heat. Okay, so it speeds up a chemical reaction without having to use heat. So I may be completely getting ahead of myself because I don't know if we're going to discuss this or not. So just in case we don't, I'm going to talk about it now. If we do come to it later, I'll just skim over it. But I want to remind you again, and this ties back into the chapter we discussed diffusion and osmosis, because it helps us to understand how these enzymes work. What we discussed back then was this Brownian movement of molecules and atoms. And that's just the natural movement that atoms and molecules undergo all of the time. There's always some motion. And remember, solids is the least, liquids is the next most, and then the highest amount of motion is gases. And that's the three major forms of matter. There is another one we don't need to discuss in here. But these molecules are always moving. And when molecules are moving, the more they're moving, the greater likelihood they have of interacting with each other, because the greater the likelihood that they will come into contact with each other. So by increasing the speed at which these molecules are moving, increases the likelihood that a reaction will occur among them or between them. So when we want to speed up a reaction, we heat it, because heat increases the movement of these molecules. I probably used this illustration, but that's why if you put sugar in iced tea, it dissolves much more slowly than when you put sugar in hot tea. Okay, the same tea, but one's hot and one's cold, because of this movement of molecules. So the problem is this: we can't heat a living organism up to, there's generally a specific range at which this organism can survive. And so oftentimes the heat that we would need to apply would be so great that it would actually kill the organism. And so in order to get these reactions to occur without heating it up to the point where it would kill it, we have to use these things called enzymes. And we will discuss how they work now. So we call the things that we're putting together reactants or substrates or products, depending on what sort of phase they're in, I guess. So we can convert these products or these things into products either by forming a bond or by breaking a bond. So the substrates are the reactant atoms or the reactant molecules that the enzyme does its thing to. So as I mentioned, okay, so we do talk about it a little bit. Enzymes speed up the rate of these reactions without increasing temperature. They're much larger than the substrates, and the most simple explanation for how these work, there are other theories, but the most simple is the one that's called the active site theory, which means that these enzymes have a site on them. And remember, they're always proteins. I don't know if I mentioned that or not. That's not true. We do have some RNA enzymes, but they're usually proteins. So they have a specific three-dimensional shape to them, and they can create hydrogen bonds and probably things like disulfide bonds and maybe even other stronger peptide or covalent bonds. I'm not exactly sure. I don't know if we even know. But they have these sites that will bind the substrate. So what this picture is showing here, obviously this is the enzyme, and here is the active site. And here are two different substrates, and you can imagine which one of these is going to fit. If you remember playing with toys when you were 3 years old, this should be obvious to you. This one's not going to fit in there, right? So this one is, and this particular enzyme is an enzyme that breaks the substrate apart. So it bonds to this active site, and then somehow this enzyme pulls apart this substrate into these two products. So that's, an example would be how we catabolize some specific type of a macromolecule. So they bind the substrate and participate directly into the changes, and as I said, it doesn't become part of the product. The enzyme stays intact when this is finished. Therefore, it's not used up, and we can use it over and over and over again. So when we talk about the speed of an enzyme, we're talking about how fast it converts the products. So an example is catalase. And this is per second. Catalase, and usually we use it per minute with anatomy and physiology and biology and things like that. But these things happen at such a massive rate. Well, that's the wrong word. A high rate that we can't even measure it in minutes, because catalase, for example, which breaks down hydrogen peroxide, because remember hydrogen peroxide is harmful. It breaks it up into oxygen and water. Catalase can do that chemical reaction several million every single second. Okay, another example, lactate dehydrogenase, which catalyzes pyruvic acid and lactic acid back and forth by using an NADH, which we'll talk about a little bit later. Anyway, it can convert this lactic acid at the 1,000 per second. So just super, super, super fast. So let's look at some of the different types of enzymes and the structure of them. So there's simple enzymes, which is nothing but just a protein, and that's what I showed you on the previous slide. And then we have these things called conjugated enzymes, also known as holoenzymes, because these things have the protein, but also a non-protein molecule. So when we add that non-protein molecule, we call the protein part of the holoenzyme the apoenzyme. So all the purple is the apoenzyme, and then this is a holoenzyme, this is a holoenzyme, this is a holoenzyme. So there's different non-protein molecules that we use. Cofactors is an example, and they can either be organic molecules that we call coenzymes. Remember, organic has carbon in it, right? Carbon and hydrogen. Or they can be inorganic elements, such as metal ions. So that's what this is showing here is a metallic cofactor. So coenzymes is a cofactor that is organic, and then a metal is a cofactor that's inorganic. And an example of where we use a cofactor is catalase. It has to have an iron as its cofactor or it doesn't function properly. And that's why we take things. This is one of the major reasons why we need to consume minerals, because minerals are often used as cofactors. So in your multivitamin, you'll probably see all kinds of things on there that are used for cofactors. So this table in your book just shows some of the major characteristics of enzymes. And we've already discussed everything on that list except the last one, can be regulated by feedback and genetic mechanisms. And all that means is, well, let's see. I know we do discuss in later chapters some of this, but feedback mechanisms, which would be usually negative feedback mechanisms, essentially this: when you create the product, it stops the enzyme from continuing to create more products. Genetic mechanisms would be genes, which we talk about in the next chapter. Genes turning on or turning off enzymes, usually in the form of creating the enzyme, because remember, an enzyme is usually a protein, and the gene has the instructions for creating the protein. We will discuss what's called the lac operon, and it'll make sense next chapter. So the enzyme-substrate interactions. So a temporary enzyme-substrate union must occur at the active site, which I already discussed, and they call this the lock and key theory, because a key goes into a lock and it's a very specific shape, and only the right key will go into the right lock. And so that's the idea behind this picture here, as I mentioned. So the bond between the enzyme and the substrate is weak, and that's really important because once we create the product, it needs to be able to be released very well and quickly, right? Once it's released, then the enzyme is able to interact with another substrate.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
So as we mentioned, these cofactors help this process along. So these trace elements or trace minerals generally are used for these cofactors. Examples: iron, copper, magnesium, manganese. So metals and usually in the form of ions, metals and minerals. Participate in the precise functions between the enzyme and the substrate by doing things like help bringing the active site and the substrate closer together. Sometimes participate right in the chemical reactions, and so on. They just do things, right? So coenzymes, as I'd mentioned, was a type of cofactor that was organic. And usually what they're there for is to move a chemical group, like a phosphate or something like that, from one molecule over to another one. So they can carry and transfer hydrogen ions or electrons, CO2, amino groups, things like that. So a transient carrier is what we call this, because a transient means that it's temporary. So a lot of these coenzymes come from vitamins. So when we eat food, we use it for energy, we use it for building blocks, and then we use vitamins for these coenzymes, which are neither building blocks nor used for energy. So we classify these enzymes according to certain characteristics that they have, such as the site of action, the type of action, the substrates that they either use or make. So the name is composed of a prefix or a stem word derived from one of these characteristics, such as what the substrate um, or which substrate they act upon, the type of reaction that they cause, and then we put an ending on it, which is ACE. So whenever you see ACE, it's an enzyme. I don't know any exceptions to that. There may be, but I don't know of any. So there's 6 classes of enzymes based on their biochemical reactions. The first class, these are called oxidoreductases. Oxidoreductases. Redox reactions, I don't remember if I think we talk about that this chapter. That's what where that comes from. Oxidation reduction reactions. So if you remember from your chemistry class, that's what we're talking about here. So what they do is transfer electrons from one substrate to another. An example is a dehydrogenase, which transfers a hydrogen, and it's odd, but we consider that to be transferring an electron because I'm not exactly positive why, but the hydrogen has an electron on it, and so they, for some reason, focus on the electron in that example. But D obviously is, taking away, and then hydrogenase is hydrogen. So transferases transfer a functional group from one substrate to another, like a phosphate group. Hydrolases break bonds by adding water. Hydro means water, and hydrolysis is breaking apart water. So that's not to be confused with hydrolase, not the same thing. It uses water but doesn't break apart water. Lyases add or remove groups to double bonded substrates. Isomerases change a substrate into an isomeric form. Now, isomers, that is also a chemistry thing. So an isomer iso means same. It's Greek, I think, Latin, Greek, I don't know, one of the two. It means same, and what that means is you can have two different molecules with the same chemical formula, meaning they have the same number of carbons and the same number of hydrogens and the same number of oxygens, yet they're shaped completely differently. So that's what an isomer is. So an isomerase will change, you just rearrange the same atoms and create a different molecule with the same atoms in it. That's what an isomer is. A ligase creates chemical bonds using the energy from ATP and by removing a water. So all of these enzymes also have a common name that indicates what the thing actually does. For example, carbohydrase digests a carbohydrate substrate. Amylase, which is in your saliva and also released from your small intestine, breaks starches apart into simple sugars. Maltase digests maltose into 2 glucose molecules. Maltose is a disaccharide. Proteinase, proteases, peptidases all break peptide bonds. Those are released in your stomach, also in your small intestine. Lipases break apart lipids. Deoxyribonucleases or DNases digest DNA. Synthetases or polymerases Polymer means chain, right? Bonds many molecules together into a chain. So how do we regulate these things? Well, there's different types of regulation that we use, and so these enzymes will have different names. The first one we're going to talk about is called constitutive enzymes. And constitutive enzymes are always present, no matter what the levels of the substrate are. And generally the reason why this is the case is because the chemical reactions that these things help with are uh, pretty much vital for the cell to be alive all of the time. Then we have regulated enzymes. Regulated enzymes can either be induced or turned on, or turned off or repressed in response to the concentrations of the substrate. So these enzymes are uh, highly influenced by what's going on around the cell, such as the temperature of the cell, pH, osmotic pressure. And part of the reason why is because these changes, these of what we consider to be normal conditions, can cause the enzymes to become very unstable, or what we call labile, such as too much heat or too low of a pH, for example. And if we change the enzyme enough, we end up denaturing the enzyme. And when you denature the enzyme, it actually changes the shape. And as you probably have heard me say, the shape of a protein determines the function. So if you change the shape, you change the function. So things like heat and pH, as I had already mentioned. And the reason why is because it starts to break bonds that hold the three-dimensional structure of the protein. And when you change the three-dimensional structure, then the enzyme can no longer attach to the substrates, and obviously it's non-functional at that point. And that's what denaturation is. So like denaturing in the proteins in an egg, for example, when you cook it, it goes from being a slimy, clear, the white part, to solid and um, opaque. So there are these things called metabolic pathways in um, the metabolism of living organisms, and there are these sequential steps, or processes of these metabolic reactions that occur. Usually multi-step series or a pathway, and each step is often catalyzed by a separate enzyme, sometimes multiple steps by the same enzyme, and there are lots of different examples. And usually the product of one reaction is one of the reactants in the next reaction. So it creates a chain, or sometimes you'll hear called a cascade. Also, in these metabolic pathways, oftentimes the metabolic pathway can branch and take a right or a left turn based upon environmental conditions. Sometimes, instead of being linear, though, these metabolic pathways are cyclical. They go around in a circle. And in those kinds of pathways, the molecule that we begin with uh, is regenerated to sort of prime the next cycle to start. So these metabolic pathways almost never stand alone. They are interconnected with other pathways and merge at many different sites along the pathway. So you don't necessarily have to start at the beginning and end at the end. Sometimes what can happen is you can start in the middle, because you can create uh, whatever the product is in the middle of the pathway via another process, and just jump right into the middle and not have to begin right at the start. So this right here just shows some different types of these metabolic pathways. So this one here is called a linear pathway. An example is glycolysis, which is where we take sugar and break it apart. So A to B to C to D, and then we end up with some sort of a product. This is a cyclic, and the example is like the Krebs cycle, also called the citric acid cycle, which is where we start here, go around in a circle, and we end at the same place we started. And as I mentioned, there can be branched ones. So this is an example of a divergent branch where N can either become O or it can become P. If it becomes P, it'll end up as an R. If it becomes O, it can end up as this thing down here. And then convergent is where two different pathways come together to create something. An example is when we create amino acids.
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
So earlier when we were looking at, what was it, table 7.1, I mentioned that there were different ways to regulate this. One was genetic and the other one was by feedback. Well, let's talk about how we can regulate those things via feedback. So one of the ways is called competitive inhibition. And this is inhibiting the function of the enzyme by putting a molecule on the enzyme that will attach to it the same way the substrate will attach to it. And what that does is it blocks the active site. So if you look over here at this picture, this is our normal substrate, and the bottom of it is shaped like this. Well, you can see that the green one has the exact same shape, and so either one of these will fit in here. So if this competitive one is present, it will block it and not allow these this catabolic reaction to occur. If it isn't present, then this normal substrate can go ahead and react with the enzyme and become these two products here. Non-competitive inhibition is when the enzyme actually has two different bonding sites. They call those allosteric molecules, and again, the lac operon that we'll talk about next chapter is a great example of this. So the two binding sites are the active site and the regulatory site. So the molecules bind to the regulatory site and change the shape of the enzyme and not allow the substrate to attach to it. And so it can either slow down or stop the activity of the enzyme. So enzymes don't last forever. Sometimes they wear out. Sometimes the organism degrades them deliberately so that there is a lifespan on them. And then some of them are diluted with each cell division. What that means is if you start with, say, 10 enzymes in a bacterium, and the bacterium undergoes binary fission and splits into two, half goes to one bacterium and the other half goes to the other bacterium. So now instead of having one cell with 10 enzymes, we have two cells with 5 each. And then if they split again, one's going to end up with 2 and one's going to end up with 3, and so on and so forth. So they just continually get diluted with each cell division cycle, each generation. So we regulate these enzymes based on the cell demand, right? Obviously, if we don't need them, we're not going to waste the energy creating them. So enzyme repression is genetic control that keeps the enzymes from being produced. So this kind of control mechanism is much slower than the feedback mechanisms, but the effects last longer. Enzyme induction, so repression is where we repress it. Enzyme induction means that the enzymes are produced via translation, which we'll talk about next chapter, only when we have the substrates present. And I keep mentioning this, but the lac operon is a perfect example of that. We only produce the enzymes that break apart lactose when the lactose is present, and also when there's no glucose present. So this shows an example of enzyme repression. So again, we'll go into detail next chapter, but here's the instructions on the DNA. We turn that into a piece of RNA. We use the RNA to create a protein, and then we're going to head down to the bottom of this. The protein folds up into some sort of a tertiary, or if it binds with another protein, a quaternary structure, and then becomes a functioning enzyme. Then we go ahead and we do our thing, right? Create whatever products we need to create. The products then bind to somewhere on the DNA, or generally it binds to the regulatory site on an enzyme and causes it to bind to the DNA, or vice versa. It could bind to the site and cause it to release from the DNA. Different examples, but somehow it interacts with the DNA and basically blocks the enzymes that are needed, the polymerase enzymes that are needed to produce a new piece of RNA. So an example is E. coli. And this actually is partially that lac operon I was talking about. I know, I'm sorry I keep mentioning that, but at least keep it in mind, because I know you don't know what it is yet, but keep it in mind so when we get to chapter 8, you can kind of go back and make some more sense of this. You know, all of these subjects sort of build on each other, and so as we're laying this foundation, it's a really good idea to be thinking back on what you've learned previously and applying that knowledge. So just anyway, keep that in mind. So when lactose is present, it produces the enzymes to help it break the lactose apart into glucose and galactose. When there is no, well, that's actually not true. Anyway, it uses lactase. When sucrose is present, it prefers sucrose, or even if just glucose is present. So it stops creating the lactose and creates sucrase to break apart the sucrose, or just breaks the glucose down and uses that, depending on what's present. So what this does, though, it allows the organism to use different nutrients. So it has preferred nutrients, but when the preferred nutrients are not present, it will begin breaking down the non-preferred nutrients. So prevents it from wasting energy because it doesn't make enzymes when there's no use for them. Which of the following mechanisms of enzyme control blocks a reaction catalyzed by an enzyme by the binding of a product to a regulatory site on the enzyme? Is it enzyme repression, competitive inhibition, enzyme induction, non-competitive inhibition, or none of those? And the answer is non-competitive inhibition, because a regulatory molecule in non-competitive inhibition binds to the regulatory site, changes the shape of the protein, which is the enzyme, and does not allow it to bind to the substrate. Every cell every living thing for that matter requires energy to be able to continue the metabolic processes that it has to undergo. Um, Although some of them use some very strange methods, we're going to focus on the most common ones. So light energy in the form of photons, chemical bonds are the other major form of energy in which we break the bonds to release the energy. So once we release that energy, generally it's stored in the form of ATP. And the analogy I use is like ATP is like a battery. And when we need the battery, we pop it in and use the energy from the battery. And then we go and throw it back on the charger and recharge it, so to speak. Earlier in one of these earlier chapters, we discussed that, I think. So only chemical energy can be used on a regular basis because oftentimes light energy and some of these other forms of energy they're just not constant. You know, if it's a cloudy day or if it's nighttime, you know, obviously light energy is not available, at least in the form of the sun. So there's two types of reactions in regards to energy. There's an exergonic reaction, which is where the reaction releases energy. And this is redundant. I know we've talked about this a lot, but this is where you break a bond and it releases energy, oftentimes stored in these high energy phosphate bonds, which is why we use ATP. Then there's also endergonic reactions, which require energy. And these endergonic reactions and exergonic reactions are often coupled together in some sort of a process, so that when the energy is released from the exergonic reaction, it's immediately put into the endergonic reaction to drive it. So earlier I had mentioned this oxidation and reduction. So we call these oxidation reduction reactions because they happen together, or redox, which is just a shortened version of reduction and oxidation put together. So we're going to start with oxidation. And this may or may not sound like what it is. It implies something is happening with oxygen, and although that is the case sometimes, it's not always the case. So the actual definition of an oxidation is the loss of electrons. So that is to say that when something loses electrons, it is oxidized. And when, of course, something loses electrons, that ends up causing a positive charge. So it could be an ion or a polyatomic ion that has a positive charge. It's been oxidized. Reduction, then, is the gain of electrons. Now, let me see if I can help you make some sense of this. When you reduce something, you make it smaller, right? And so when you reduce chemically, you make it more negative. And that's kind of, if you can sort of imagine in your mind that making something more negative is sort of like making it smaller. I mean, it's not, but, you know, I guess you can sort of use your imagination to kind of make that make sense in your brain. But anyway, when it's reduced, then it's gaining electrons. So becoming a negatively charged ion or polyatomic ion would be an example. So an oxidoreductase is an enzyme that takes electrons from something and adds them to something else. So what they use as carriers are things like NAD, which is nicotinamide adenine dinucleotide, which is a nucleic acid, or flavin adenine dinucleotide, which is also a nucleic acid, so FAD or NAD. We'll show you some reactions where we use hydrogen to carry the electron. So a redox pair then are the two things actually involved in the reaction. And so the most commonly used example for this that we use when we talk about ions or redox reactions is sodium chloride. So clear back when we talked about ions back in junior high, we use this as an example. So the sodium is on the left side of the periodic table, so of course it's going to lose an electron. Chloride is on, or chlorine I should say, is on the right side, so it's going to gain an electron and become chloride ion. And so the reducing agent is the sodium, and the reducing agent is what is oxidized. Okay, this is totally confusing, I know. The oxidizing agent is the thing that is reduced. So it's sort of like the chloride oxidizes the sodium and the sodium reduces the chloride, because the sodium loses an electron and becomes positively charged, which is a cation. The chlorine gains an electron and becomes a negatively charged chloride. And then, of course, you know that when we have a positive and a negative ion, they can come together and create an ionic bond, but that has nothing to do with a redox reaction. The redox reaction is the movement of the electron. But the point of this mainly is that the sodium and the chloride is a redox pair. So the energy present in the electron acceptor, which in that case would be the chlorine, can be captured to phosphorylate an ADP or some other compound like a creatine phosphate or creatine phosphate or something like that. So the handling of electrons then, in some cases, the cell doesn't handle the electrons as just a separate electron, but instead carries it combined with another atom like a hydrogen. So it's very odd because when we talk about sending a hydrogen, for example, hydrogenating an NAD, we talk about it as reducing the NAD because it's carrying an electron with it. So it doesn't technically even give it a charge, which is kind of confusing. So dehydrogenating then is the removal of a hydrogen during this redox reaction. And that transfers one electron because a hydrogen has one proton, therefore it also has one electron, right? And remember, most hydrogen exists as protium in the world, so there's no neutron, there's just one proton and one electron. And so this gives us an example of an electron carrier. So your book calls these things molecular shuttles. So, and that's the reason why is because the carrier shuttles the electron. So it loads one and then it unloads it, and then it loads another one and unloads it, and continues to do this repeatedly to be able to facilitate this reaction occurring. So during aerobic metabolism, air meaning air, which denotes oxygen, the final electron acceptor is oxygen. In anaerobic metabolism, it's something else, like CO2 or something like that. So the picture that we're looking at here shows an NAD, nicotinamide adenine dinucleotide. And it has a positive charge, right? So when we add two hydrogens, what happens is, because NAD is positively charged, one of the electrons from the two hydrogens, because they're carrying two electrons, right? One of the electrons and one of the hydrogens with the other electron combines with this NAD+. And then that leaves a free proton right here. And that's why we end up with the reaction NAD+ plus two H's goes to NADH plus the H plus that's left over. This is the proton. And then this picture right here just shows what's happening. So the hydrogen bonds at this carbon right here, and then the electron, of course, is just orbiting around as in the cloud, right? And then this other proton that's left over is not attached anywhere. It's just released as a free radical. So ATP is something we will talk about forever in any subject regarding biology. So let's talk about what it is. We've never taken a really close look at it yet. So it's a three-part molecule. It's got a nitrogenous base, which is adenine. That's the A in ATP, adenine or adenosine triphosphate, right? A 5-carbon sugar, which in this case is ribose, just like in RNA, which was ribonucleic acid. So it's the same 5-carbon sugar, which is right here. So here's the adenine. It is a type of molecule called a purine. We'll talk about a little bit later in the next chapter, I guess. So we have the adenine, the 5-carbon sugar, which is ribose, and then we have a chain of these 3 phosphate groups. So PO4 minus. So the phosphate group, and we call these things phosphates a lot of times, and that implies that it's a phosphate atom. But I mentioned this somewhere else, I think, but it's not a phosphate atom, it's a phosphate group, which is a phosphate with 4 oxygens and a negative charge. Okay, so here's the phosphate, 1, 2, 3, 4 oxygens. Anyway, the other way we write phosphate is P with a little I at the bottom, and this I is usually italicized. No, it's not italicized. It's usually one of those little squiggly eyes is what I meant to say. Anyway, so this the chain of these 3 phosphates is the third part of this. So the phosphate groups are bulky and carry a negative charge, as it says right here. And what that does is it causes there to be a strain between the last 2 phosphates. So when we remove one of those phosphates, it releases energy. And when we do that, we take apart the ATP, which is with the 3 phosphates, and what we end up with is a DP, which only has 2 phosphates. So when we break that bond right there, we end up with only ATP. And there's more to it than that because it actually removes another one, and then we have to put it back together. But we don't need to worry about that. Just remember that it takes off the last one at this point. So the ATP utilization, meaning we break the bond of the last phosphate group and then putting it back on, is an ongoing cycle. We're constantly breaking that phosphate off and putting it back on. So it's hydrolysis, which is the chemical reaction that occurs to break it off. And this is what powers almost everything in biology. And so we use enzymes to put this molecule together. And there's all kinds of functions, I guess one of which is for catabolism. So in a redox reaction, loss of electrons is phosphorylation, oxidation, fermentation, or reduction. And we know that reduction is reducing or gaining electrons. Therefore, oxidation has to be the opposite of that, which is the loss of electrons. So the answer is B. So catabolism, breaking things down. As that table at the beginning of the book showed, we use enzymes to catabolize things down into precursor molecules that we then can use as building blocks to anabolize into the complex molecules that we need. And oftentimes we break something down from what we need it. Let me start over. Let's say you need a piece of collagen. Well, when you eat a piece of meat, there's collagen in it. But we have no way of taking collagen and implementing it into our tissues. So we actually have to break the collagen apart and then reform it into collagen again. So oftentimes, I guess my point is we're building the very thing that we're breaking down on the front end, but that's just because that's the only way we can actually build it the way we need it. So there's this thing called reducing power. And we have to use this reducing power for these things to occur. So we have these electrons available in NADH, which was the reaction that I had showed you earlier, where we take an NAD plus and add 2 hydrogens and end up with NADH and a free proton. FAD works the same way. So the energy that we use is stored in these bonds of these molecules, namely ATP. And we need both reducing power and the energy in large quantities for these metabolic reactions to occur. And both of the reducing power and the energy is produced during catabolism of our food.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Here's the 3 main metabolic pathways, and we're going to talk about all of these. Aerobic respiration is what aerobic organisms use, such as humans, animals. That's the one obviously on the left. The final electron acceptor is oxygen. Anaerobic respiration, very, very similar. There's just a couple of differences, and the main one being that the final electron acceptor is not oxygen. That says non-oxygen there. Look at the yielding of ATPs. It depends on where you end up creating or where you stop the cycle with anaerobic respiration. But 36 to 38 ATPs here, 2 to 36 here, and in fermentation, 2 ATPs is all that's produced. So let's go through the details of every single one of those. So the way bacteria process the food that they eat varies dramatically. In most cases, and this is what we'll focus on, the energy producing nutrient is glucose. So aerobic respiration converts glucose into carbon dioxide, and remember the final electron acceptor is oxygen. That's why aerobic respiring organisms require oxygen and give off carbon dioxide as a waste product. And when we break these molecules down, we get just a very high number of ATPs. So some of the processes that occur in aerobic respiration, because it's multiple processes, glycolysis, the Krebs cycle, and the electron transport chain are the 3 major processes. So as I'd mentioned, free oxygen is the final acceptor, and this is how many bacteria, all fungi, as far as I know at least, protozoa, and all animals respire. Anaerobic respiration, used strictly by anaerobic organisms because they can metabolize without the oxygen, still involves glycolysis in the Krebs cycle and the electron transport chain, but it uses something like nitrate, sulfate, or carbonate is what these are. So these non-oxygen. Fermentation is the last one, which is incomplete oxidation of glucose. So it begins to oxidize it, but it doesn't continue on through, and that's why it doesn't produce as many ATPs. Oxygen is not required, so it uses some sort of an organic compound as the final electron acceptor, carbon and hydrogen containing compound. So this is glycolysis. So remember, there was those three basic processes: glycolysis, Krebs cycle, and the electron transport chain. So this is the first one, glycolysis. So a glucose molecule is C6H12O6. So it's got 6 carbons, 12 hydrogens, and 6 oxygens. So during glycolysis, glyc means sugar, lysis means to break apart. So when we lyse sugar, right, we're breaking apart a glucose. And the first thing that happens is glucose is converted to fructose 1,6-diphosphate, which is just a different type of 6-carbon sugar. Then we take the fructose 1,6-diphosphate and break it apart into 2 3-carbon sugars called, well, I don't know what they're called, but then they're converted into pyruvic acid, which is this right here. Now in this step, notice that we yield, because it's a decomposition reaction, 4 ATPs and 2 NADHs, which are the reducing agents, right? The reducing power. The problem is we used 2 ATPs up here to convert it. And so our net yield is 2 ATPs and 2 NADHs. And we end up with these 2 pyruvates, or pyruvic acids. And that's glycolysis. So from there, we're going to run these pyruvic acids through the Krebs cycle.
Cells derive energy from the oxidation of nutrients such as glucose. The oxidation of glucose to pyruvate occurs through a series of steps called glycolysis. The energy released during these oxidation reactions is used to form adenosine triphosphate, ATP, the energy currency of the cell. The initial steps in glycolysis are the additions of 2 phosphates to the glucose molecule at the expense of 2 molecules of ATP. The result is a 6-carbon sugar diphosphate molecule and 2 low-energy adenosine diphosphate molecules, or ADP. This 6-carbon sugar diphosphate molecule is then split into 2 3-carbon molecules. Each of the 3-carbon molecules is converted through a series of steps to pyruvate. During these reactions, electrons are transferred to the coenzyme NAD+ to form NADH, and ATP is formed. Under aerobic conditions, the pyruvate is further oxidized to yield more ATP, and under anaerobic conditions, the pyruvate is converted into lactic acid.
So once we have made our 2 pyruvic acids, we run the pyruvate through the Krebs cycle. Now notice there's 2 pyruvates here. We're only going to show it just running through one of them, but bear in mind each of the pyruvates will run through this. So whatever we end up yielding, you just have to double it, right? So the reason why we continue this is because this pyruvic acid is still very energy rich. There's still bonds that we can break apart, right? So it's important that you understand where this all happens. In bacteria, prokaryotes, this occurs in the cytoplasm because they don't have organelles, right? In the eukaryotes, it occurs in the mitochondria, which, if you recall, is that double membrane, the powerhouse of the cell. And so these reactions occur back and forth on either side of the membrane, on the inner membrane, in those folds called cristae. I think we show a picture of that a little bit later. So it's a cyclical metabolic pathway, just meaning that it runs in a circle and begins with something called acetyl-CoA. So the acetyl-CoA joins with this oxaloacetate or oxaloacetic acid and forms a 6-carbon molecule called citric acid or citrate. And that's why the other name for the Krebs cycle that you often hear is the citric acid cycle. So it continues on around this Krebs cycle, and there's certainly more detail than what we're going to go into in this class, but it continues on around here. And as it continues, it goes through several more steps, and along the way it begins releasing different forms of energy like guanine triphosphate or ATP. I'm not entirely sure. Sometimes it's GTP, sometimes it's ATP, and I think it just happens to be whatever is available, but I'm not positive about that. But it doesn't matter. GTP and ATP, essentially, for all intents and purposes, they function the same way. But eventually, once we run it through this, we end up back here at this oxaloacetic acid. And notice we had 6 carbons to begin with. We only end up with 4. Well, 1 got lost here, where we combined it with an oxygen. So we formed 1 CO2 here, and then 1 we lost over here when we combined with another oxygen. And this is why we require oxygen for this process to occur. And this is why we give off carbon dioxide as a waste product. So then once we're finished, we're back to this oxaloacetic acid, which we combine again with another acetyl-CoA, and we just run it again. And this, you can see, yields 3 NADHs and 1 FADH2. So we transfer that energy into the NADH and the FAD by reducing them, by adding the hydrogens, which remember are carrying electrons with them, right? So then we take these reduced NAD and FADs, which are now NADH and FADH2, and we then send them through the electron transport chain. So 2 ATPs are produced for each molecule of glucose run through this process.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
So that brings us to the last part of this process called the respiratory chain, or the electron transport chain. Called that because we're transporting electrons from one molecule to another. So it's this chain of these redox carriers that receives the reduced carriers, which remember we reduced in the Krebs cycle in the form of NADH and FADH2, and sends it through these different carriers sequentially and produces energy on the back end. And it's carried, these electrons, remember, are carried in the form of these hydrogen ions that are attached to the NAD plus and the FAD, right? So there and there. So in the final step, oxygen accepts the electrons and the hydrogen, and we create water. So the actual byproducts of this whole process is, or are, I should say, water and carbon dioxide, and then also energy is released. So the principal compounds in this whole process are NADH dehydrogenase, which is an enzyme. These things called flavoproteins, which are embedded in the membrane, and then coenzyme Q, also known as ubiquinone, and cytochromes. So cytochromes are these enzymes or these carriers, I guess, that are embedded in the membrane of the either the cell membrane in a prokaryote or in the inner membrane of the mitochondria of a eukaryote. And that's what these things are right here. It shows 5 of them, but they can have more than 5. Okay, so we start with our NADH, and we can, and just bear in mind we run the FADH2 through this same sort of process, but we're just going to talk about this one. So we start with our NADH right here. That NADH then is broken apart using the NADH dehydrogenase. That releases protons off of the NADH. Okay, so an H+, which is a hydrogen ion. Those things then diffuse, or rather are transported out of the cell, and so we end up with this concentration gradient where we have a lot of protons on the outside. And that creates a condition of acidity. So the pH is actually lower outside the cell, and that makes it more acidic. And so this creates what's called this proton motive force, and that causes these protons to flow down the concentration gradient, right? Because they always want to go from a concentration of high to low. And when they do that, it fires off this ATP synthase enzyme, which is right here. And when that happens, so we're diffusing these out through, and that the energy that is created from that is the energy that we create an ATP with. So we take an ADP and a phosphate group, and we add them together to make a phosphate. And then we end up with all these protons back inside the cell here, or inside of the inner membrane. And at that point, we need to do something with them. So we do things such as combine them with oxygen to make a water, or combine them with nitrate to make oxidized nitrate, or sulfate to make, what is that, hydrogen sulfide. No, that's actually not true. That's bisulfide, HS minus, not hydrogen sulfide. Hydrogen sulfide is H2S. So sorry, ignore what I said. But the whole point of this then is we create several ATPs doing this. So every single NADH creates 3 ATPs, and every FADH creates 2 ATPs. So as I had mentioned in eukaryotes, this all happens on the inner membrane of the mitochondria. So here's the mitochondria. These folds are called cristae, and then this is a zoomed in view. So this right here is the ATP synthase, this brown one. So that binding of this ATP synthase on the end of this electron transport chain, that's what we call oxidative phosphorylation, because we're using the oxidative power to phosphorylate or put a phosphor on an ADP, right? And as I'd mentioned on the previous slide, We get 3 ATPs out of an NADH and 2 out of an FADH. So the terminal step, the last step, in aerobic respiration, it is catalyzed by cytochrome AA3, also known as cytochrome oxidase. And that was where I showed the free hydrogen binding to oxygens to create water. And this is the actual chemical reaction of it. So we have these two protons that run through the ATP synthase and that forms the ATPs, and we then combine them with free electrons and an oxygen. It says half oxygen because oxygen exists as O2. So we take 4 of these and 1 O2 molecule, and what we end up with is water. So in the case of this one, it's 1 water. If we did what I said earlier, it would be 2 waters. But anyway, you get the point. We take the protons, add them to oxygens, and we end up with water as a byproduct, which is obviously something we can use. So most eukaryotes have this cytochrome system that runs through this whole entire process. Bacteria can have all kinds of variations on this, though. They can have one or more steps be missing, or different types of electron transport schemes that run these electrons through this system. And one very important thing is some of these don't have cytochrome C oxidase, and that's what allows us to separate different genera of bacteria and differentiate one from another. So one of the things that can happen is we don't end up creating the water and the carbon dioxide like we showed. And we can end up creating either a superoxide ion, which is very harmful, or a hydrogen peroxide ion, which is also harmful. And so we had mentioned earlier in one of the previous chapters how we deal with those things. But we're going to talk about it again. So we have enzymes that are made to deal with these toxic oxygen products. One of them is superoxide dismutase, which converts the superoxide ion, and then catalase converts the hydrogen peroxide into oxygen and water. So Streptococcus species lack these enzymes, but they're still able to grow in oxygen because they produce the enzyme called peroxidase.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
So remember, this happens in the inner membrane of mitochondrion, which is what that's showing there, but don't forget it also occurs in the cell membrane of prokaryotes that do not have mitochondria. So the terminal step, so that was our aerobic respiration. So the terminal step of anaerobic respiration doesn't use oxygen as the final electron acceptor, right? So it's either nitrate or nitrite, which is reduced nitrate or oxidized nitrate, right? Those are the two most common ones. So this shows the nitrate reductase system. So we have this electron acceptor, which is nitrate right here, and we release the hydrogen off of this NAD, right? And what we end up with on the other side is a nitrite, reduced nitrate, and a water. And then when we take the hydrogen, off of this and the extra electron, we end up with a back to our NAD plus again. So denitrification is something that occurs in certain species like Pseudomonas and Bacillus. They can actually reduce the nitrite. So most can reduce nitrate into nitrite, but some can even further reduce nitrite into nitric oxide, NO, or nitrous oxide, which is actually laughing gas, which is N2O. Or even produce nitrogen gas. And that's a really important thing because a lot of organisms don't have the ability to do this, but they also do require nitrogen. And so we use this relationship with these bacteria to sort of do the job for us. Other examples besides nitrate and nitrite are carbonates and sulfates. Aerobic pathways always produce the most ATPs, with anaerobic pathways being next, and then the least of which is fermentation. So fermentation, as I said, is the incomplete oxidation of glucose or other carbohydrates, just other sugars, when we don't have oxygen. So some organic compound is the terminal electron acceptor instead of oxygen or these nitrates, sulfates, or carbonates. And it yields a very small amount of ATP by comparison. So this is the type of system that's used by organisms that don't have an electron transport chain. Or in organisms that when oxygen is not present, they don't waste the energy creating the electron transport chain, enzymes and proteins. So it only yields 2 ATPs per molecule of glucose. And in spite of this process not being as efficient, many bacteria can grow at the same rate that they could if they were in an aerobic environment, because they can actually speed up the rate of glycolysis, breaking apart that glucose or other sugars into like pyruvate or whatever, lactic acid or whatever. So it permits the independence from molecular oxygen, meaning they don't need the oxygen to be able to do this. And this is why we can have anaerobic bacteria or various levels of oxygen concentrations and still be able to grow. So one symbiotic relationship where this is important is with bacteria and ruminant cattle. Ruminants are these animals that have more than one stomach, like cows. And in these stomachs, they have these bacteria living in them that digest the cellulose through the process of fermentation. So they take, and remember cellulose, which is the cell wall of the plant, or in other words, it's wood. Cellulose is hydrolyzed into glucose because it's actually made up of glucose, but the animals don't have the enzymes required to break the bonds to release the glucose. So when the glucose is fermented, it produces different organic acids that then are absorbed into the cow to give it its energy. So human muscle cells can undergo a form of fermentation. So when oxygen levels are depleted, which actually doesn't take very long, we can still continue to use our muscles. And the way we do it is by converting pyruvate into lactic acid. So instead of running the lactic acid through the Krebs cycle, we run, pyruvate through the Krebs cycle, we run lactic acid, and it doesn't require oxygen. So we can still gain our ATPs without the use of oxygen. The problem is we end up producing a lot of this lactic acid, and it causes the muscle to fatigue. And it used to be thought that this is what makes a person sore afterwards, but we know now that it actually isn't. This lactic acid should be gone from your body within an hour or so of exercising. So this shows two possible choices. Now this is based on what animal does what, okay? If it happens to be a fermenting bacterium, it's going to run it down this left-hand side and produce acetaldehyde, and that will be converted into ethyl alcohol, and it's going to create ATPs when we release this hydrogen and electron off of this NADH. And so we end up with this NAD plus at the end. This is how we make beer and whatnot. A human will go down this side, and we will produce lactic acid, not ethyl alcohol. That's why we don't get drunk when we exercise too much. Maybe just afterwards. All right, so what are the things that we use this for? Alcoholic beverages, because it produces ethanol, which is the kind of alcohol that we drink, and carbon dioxide. That's why beer is bubbly. We can use it to produce solvents like acetone or butanol, organic acids like lactic acid or acetic acid, vitamins, antibiotics, hormones, all kinds of stuff. And so they'll use this in industrial settings to produce all kinds of these things on just a very large scale. So we've been talking about how we catabolize glucose, but what about other non-carbohydrate compounds like lipids? So the polysaccharides is what we talked about earlier. So we can take large sugars like polysaccharides, starches, things like that, break them down into simple sugars and run the glucose through the cycle. But we can also break things down like lipids. So lipases break down the lipids into glycerol and fatty acids. And then glycerol, which is a 3-carbon molecule, is converted into dihydroxyacetone phosphate, which can then jump into this glycolysis pathway midway and not to start at the beginning. The fatty acid tails then, so if it's a triglyceride, it's going to have 3 fatty acid tails, will undergo what's called beta oxidation, and fatty acids are also carbon hydrogen molecules. And so after beta oxidation, what the products of beta oxidation can enter into the Krebs cycle as acetyl-CoA, which you may recall was that 2-carbon molecule that we created out of the pyruvic acid. So proteins are broken down by enzymes called proteases, and we have these, in in humans and animals in the digestive tract. But the amino groups are taken off of these amino acids in a process called deamination, and this, by the way, is the primary reason we have urine, is to get rid of these nitrogenous waste products from protein metabolism and nucleic acid metabolism and so on. So what's left after we take off the amino groups is these carbon compounds that we then can convert and jump back into the Krebs cycle and break them further apart to create energy from. So what is the maximum net yield of ATP per molecule of glucose for each of the following types of respiration? Aerobic, anaerobic, and fermentation. So if you go through and you, which you should, go through and add all of these processes up, glycolysis, the Krebs cycle, the electron transport chain in both aerobic and anaerobic respiration, as well as fermentation, you're going to come up with these numbers. So for aerobic respiration, somewhere between 36 and 38 ATPs are produced per molecule of glucose. For anaerobic respiration, it's somewhere between 2 and 36. And for fermentation, it's 2 ATPs. So your book uses this term, the frugality of the cell. And what it's trying to talk about is just the fact that the cell is always trying to save as much energy as possible. They don't waste. So all these catabolic pathways that we have all have these points at them where we can divert the intermediates into other processes depending on what is needed. So in other words, they can have multiple purposes depending on what the circumstances are. And so this allows these organisms to get the maximum benefit out of them. So there is this term called, what your book, they pronounce it Amphibolism, but Amphibolism or Amphibolism or something along those lines. Amphi means two. This is the ability of the system to integrate both the catabolism and the anabolism together at the same time to improve the efficiency. So this table right here is what that's trying to show. And what this is trying to explain is the back and forth that can occur depending on what is needed. For example, let's say we're running a glucose through and we're going to head it over here through the Krebs cycle to create an ATP. But partway through, once we hit glycolysis, we realize, well, hey, we need this over here somewhere else to make a carbohydrate or to create an amino acid. What we're going to do is we're going to still, like, say, I don't know, a pyruvate or something. And add it to this amino group and create an amino acid, right? Or create some sort of a carbohydrate or whatever. And that can happen obviously here too, where we take acetyl-CoA and convert it into some sort of a fat. Well, it goes both ways too. Rather than having to start at the glucose every single time, we can take, because of beta oxidation, take part of the fatty acid and we can drop it right in here to the acetyl-CoA and skip glycolysis and go straight into the Krebs cycle. And so you can see this is happening on all different levels of this. So as we've mentioned before, catabolism and anabolism. Remember, anabolism is forming bonds, right? So there's two possible sources for these macromolecules, which include monosaccharides, amino acids, fatty acids, nitrogen bases, vitamins, and so on. They could either enter the cell from the outside as nutrients, or can be synthesized from various pathways that and that was what we were just showing on the previous slide. So the degree to which an organism can synthesize its own building blocks is genetically determined, and varies from group to group. So autotrophs only require carbon dioxide as a carbon source and a few minerals, and they can make anything that they want. Some heterotrophs, like E. coli, can synthesize all of the substances from just a few minerals and one organic carbon source, like glucose. And we could go on and on and on and on about this. Point is, there's a whole bunch of different ways. So how do we synthesize carbohydrates? So as we've mentioned over and over and over, we obviously need glucose, right? Besides being for energy, it's also a major component in plants, at least for the cell walls. We also use it to store energy in the form of glycogen or starch. It's an intermediary in glycolysis. So glucose 6P is used to form glycogen. We take the glucose, add a phosphorus to it on the 6th carbon, or a phosphate group to the 6th carbon, and form this glucose 6P, and then we can put those together into glycogen. We can also take that, or we can create fructose 6P and link it to amino acids and create a peptidoglycan. We use ribose and deoxyribose in forming ATP, DNA, and RNA. We use types of polysaccharides as capsules and the glycocalyx in prokaryotes, so all kinds of different things. Proteins we use for all kinds of stuff, enzymes and carriers and all kinds of things like that, cytoskeleton and so on. So there's 20 amino acids, and those are all of the amino acids that we use to create these things. So some organisms can make all 20, which means for E. coli, there are no essential amino acids because they can create all 20 of them. For humans, it's 12 essential amino acids that we can't, so 12 out of the 20 we can't create. Other animals have to synthesize them or get them from their food because they can't synthesize them. Nucleic acids are used in DNA and RNA as well as ATP. And I should say they are DNA and RNA and ATP. DNA and RNA are both responsible for the hereditary material in viruses, prokaryotes, and eukaryotes, and we'll talk about that in detail next chapter. So, as part of this anabolism, we're always needing to replace and build cell parts, right? So both the catabolism and the anabolism are going on when the cell is in its normal vegetative state. Cell division takes place when anabolism produces enough of these macromolecules for 2 cells. Once that happens, it divides and creates 2 identical cells. So DNA replication has to first duplicate the genetic material, and then we have to double everything and then split them up. It's catabolism that provides the energy as well as the building blocks and the coenzymes for all of this to happen. So that's the reason why we need food. The ability of a cell to integrate molecule using and molecule building pathways to improve cell efficiency is known as anabolism, amphibolism, catabolism, metabolism, or none. And the integration of anabolism and catabolism, as we had mentioned earlier, was called amphibolism. And that is the end of chapter 7.